Enzymes are increasingly used during downstream processing for post-translational modification of therapeutic proteins. Monitoring the removal of such enzymes is of great importance and in most cases a requirement from FDA and EMA. One of our clients is a European biopharmaceutical company who uses 3 enzymes of human, bovine and chicken origin to modify their recombinant protein product. The client was unable to find commercial ELISA kits capable of detecting the added enzymes. Since their drug has a chance of being first to market, they did not wish to delay clinical trials by years to develop 3 process-specific ELISAs. Instead, AlphaLyse developed and validated a targeted LC-MS assay using MRM within the client's original project timeline. This analysis can be multiplexed, allowing measurement of up to 20 individual proteins in the same LC-MS run. This way, we can demonstrate clearance of all the client's process-related enzymes with a single validated assay. As part of the GMP validation, we established calibration curves for the enzyme. Each curve demonstrates excellent linear correlation covering the client's specified measurement range from 1 to 2000 ppm. This reproducible technique provides the identity and absolute amount of each enzyme, and the client now uses the analysis for batch release testing under GMP.